Thank you. I'm Sandun Rajapaksa. I'm a third-year PhD student from the Department of Data Science and AI of Monash University. Today, I'm going to talk about a part of my PhD research work which involves investigating the reliability and the limits of inference of amino acid sequence alignments.
I'm sorry to interrupt you, but can you please make it in presenter view? Like just maximize the presentation. Because we can see like the not in presenter view.
Oh, okay. Is it okay now?
Not yet. We still see all the slides on the left. Okay, if you cannot. Okay, we are fine with that. We can go with it with the left slides.
Can you see the animations as well? In my end, it's showing as a
Can you go to the next?
correctly.
What's happening? Like if it goes to the next slide or not?
There are animations. Can you see that?
No, can you go to the next slide? We can't see anything except the structure under the structure point of view, but nothing under the sequence point of view, except the sequence for sure. Yeah.
Okay. So I'll start from the beginning. An alignment is an assignment of residue-residue correspondence between pairs of proteins. It gives you a hypothesis of the relationship between proteins at the level of amino acid residues. These alignments can be based solely on their sequences or their structures. For instance, what you see here is two alignment hypotheses between human and bacterial globins using the sequence information in the left-hand side and structure information in the right-hand side. It's well known that structures change more conservatively than sequence during protein evolution. Therefore, structure alignments are more reliable than sequences. The goal of this study is to understand how reliable these sequence alignment relationships are, and also to understand the limits of inference of sequence alignments. Several studies have analyzed various regions of sequence relationships before. Commonly, these regions are categorized as daylight zone, twilight zone, and midnight zone. In the daylight zone, sequence alignments are highly consistent with the structure alignments. In the twilight zone, any single statement of a residue-residue correspondence becomes highly untrustworthy. In the midnight zone, sequences have mutated beyond any feasible recognition. In those studies, these regions were assessed using the notion of a single best alignment. The problem is it's difficult to measure the single best alignment when sequences diverge. So, if you rely on a single best alignment to study these regions, it's likely to be error-prone. In this work, we actually want to consider the fading relationship between proteins while measuring the limits of these regions. Our work builds on a Bayesian framework for protein sequence comparison called seq-malina. This framework allows you to infer sequence alignment relationship between a pair of protein sequences using a stochastic Markov matrix of amino acid substitution and associated alignment parameters for the 3 states match, insert, and delete. For any given pair, it allows you to compute a few things.
Yeah, I'm really sorry to interrupt you again, but you are stuck on slide 2 and nothing is changing. So we cannot see any change in the slides, in the platform and here in the hall. So can you please make sure that you are presenting your screen correctly?
Okay.
One option, you can select share all screen instead of the PowerPoint, and let's try to see how this will work, but try to make, to close your video, just to make sure that there is no problem. Okay, can you? Yes, it's working right now. Can you go to the next slide? Yes, everything is working. You're okay. Thank you.
Okay, thank you. I'll continue from the beginning here. Sorry about the inconvenience. Our work builds on a Bayesian framework for protein sequence comparison called seqmaliner. And this framework allows you to infer sequence alignment relationships between a pair of protein sequences using a stochastic Markov matrix of amino acid substitution and associated alignment 3-state parameters for match, insert, and delete states. For any given pair, this allows you to compute a few things. For instance, you can get the best alignment under the minimum message length framework. It also automatically infers how far the sequences have diverged by statistically estimating the time parameter associated with the underlying stochastic Markov matrix. This time parameter can be converted to a theoretical expectation of the percentage of amino acids that can change under that model of relationship. Well, the problem already stated on the previous slide is that as sequences diverge further and further, meaning that Markov time parameter grows higher and higher, a single alignment relationship over sequences, including the best one under the model, becomes less and less trustworthy. A significant property of seqmaliner framework is that it additionally allows you to infer the relationship between two sequences in an unspecified way. This is done using the notion of marginal probability over all possible alignments between the protein sequence pair. What you see here is a marginal alignment landscape where each cell ij gives the negative log of the probability that the prefixes being related and the suffixes being related. The dark blue regions show the relative high probability regions of sequence relationship between the two proteins. Associated with this landscape is an unbiased Markov time parameter. Previously, it was based on just a single alignment, but here it is basically over all possible relationships. Although the alignment relationship remains unspecified here, it gives you a lot of information about the overall relationship between the sequences. Our goal is to understand the fidelity of sequence alignments with respect to their corresponding structure alignments as a function of time. Instead of relying on a single best sequence alignment, we want to use the entire marginal landscape to compute the expected alignment distance between sequence alignments and structure alignments. To get to the expected alignment distance, you first need a measure of how distant any two alignments are. We introduce a natural and intuitive distance measure between any two given alignments between a protein pair. Any alignment can be visualized as a source to sink path in a matrix as shown here. Therefore, we can measure the distance between these two alignments by computing the amount of space or the area between the two paths. Notice that any alignment path crosses each skew diagonal of the matrix exactly once. Therefore, we can approximate the space between the two alignments by summing the width between the two paths at each skew diagonal. This width is simply the absolute difference between the corresponding proper diagonal indices of the matrix as shown in the equation here. So we can use this measure to compute the distance between any two alignments. Our goal is to compute the expected interalignment distance between the marginal landscape and a reference alignment using this measure of distance. What you see in different colors here are essentially the probability space of relationships of all possible sequence alignments for a specified pair of proteins. There is just one reference alignment in red. Let's say it's a structural alignment. We want to find the expected alignment distance between this red line and the entire landscape. What you see on the right is the exact mathematical expectation of the distance of the sequence alignment with respect to the reference structure alignment, derived by integrating over all possible sequence alignments weighted by their respective posterior probabilities. Even though the number of total possible alignments grows factorially large for any pair of sequences, we provide an exact quadratic time algorithm to calculate this. Here, we have the dynamic programming recurrences using 3 subproblem matrices corresponding to each alignment state: match, insert, and delete. Given a reference alignment, these matrices are remembering the expected alignment distances of all possible prefixes of the sequences. Let's consider the subproblem matrix for match state. The ijth cell of this matrix stores the expected distances of all possible sequence alignments ending at this cell in a match weighted by their probabilities. All the alignments that ends at the match state at cell ij are derived by extending all the alignments arriving at the cell i-1, j-1 in any of the states match, insert, and delete by a pair of matched amino acids. This extending alignment path crosses 2 skew diagonals as shown here. Therefore, for all the alignments going from i-1, j-1 cell to ij cell, the distance term at previous cell get added by this extra distance with respect to the reference alignment. By extending this distance weighted by their probability term for each state separately, we can come up with this recurrence for match state. We can follow a similar derivation for the recurrences of insert state and then for delete state. Given a pair of proteins, we now have a method to compute the expected alignment distance over all possible sequence alignments with respect to its structural alignment. This measure of computing expected alignment distance can be carried out more comprehensively. To achieve this, we randomly sampled 1 million distinct pairs of protein domains from all classes of scope. Then, we align the structures of these million pairs separately using 3 popular structure alignment programs: DALI, TM-Align, and Malina. These provided distinct sets of reference structure alignments. We also align the sequences of the same million domain pairs using seq-Malina. To evaluate the effect of changing the time parameterized models, we used 4 different stochastic Markov matrices: BLOSSOM, VTML, MML-SUM, and PAM. Then, we ran our dynamic programming algorithm and computed the mathematical expectation of interalignment distance for all these combinations. Recall that associated with each pair is the inferred Markov time. This allows us to bin the computed distances over million pairs into their corresponding Markov time bins. Then, for each bin, first, second, and third quartile statistics were computed. Finally, we could visualize the variation of the expected interalignment distance versus the Markov time for all these combinations of stochastic matrices and reference structure alignments. Results of 2 of these are shown here. The bottom axis represents the time parameter associated with the stochastic Markov matrix used. Associated with this time parameter is the expected amino acid change as shown in the top here. The left axis tracks the expected interalignment distance. Here, we have three scatter plots in magenta, green, and blue, which tracks the changes of first, second, and third quartile statistics of the expected distance over million domain pairs. The right axis tracks the cumulative percentage growth of the number of domain pairs as a function of time, as shown by the brown curve here. We have done this with various stochastic Markov matrices, and I'm presenting a couple of them here. Independent of the models and reference alignments used, a consistent pattern is visible here. For all domain pairs with time less than 150, structure alignments agree very closely. This is evident by the low value of the expected distance and the low variance between the quartile trends. This denotes the daylight zone of inference of sequence alignments. This corresponds to pairs of sequences that have undergone an expected amino acid change less than 72%. In the range 150 to 250, both the expected distance and the variance between the quartiles ramps up steeply. This specifies the twilight zone of inference of sequence alignments. This region corresponds to an expected amino acid change in the range 72% to 84% approximately. Finally, we observe the midnight zone of protein sequence relationships above 250. This region is marked by a drastic growth in the variance between the quartiles. This corresponds to a region where sequences have undergone an expected amino acid change of more than 84%. There is another analysis we perform that complements these observation of daylight, twilight, and midnight zones of sequence relationship. We grouped our million domain pairs into three groups based on the respective statistical test reported by SigmaLiner. First group contains domain pairs in which the optimal alignments are reliable. Second group contains pairs in which an unspecified relationship over the marginal results in a positive compression, but their single best alignments don't. The final set contains pairs where both the optimal alignments and the marginal probabilities are not able to provide evidence of a relationship only from the sequence information. But the relationship is verified from their classification in scope and their structural alignment. Then we plot the variation of the interalignment distance versus Markov time for each of these groups. Here we have these plots. In the first plot, we have 64% of the domain pairs in the twilight zone and another 36% within the twilight zone. This is consistent with the observation that their single optimal alignments are reliable. In the middle plot, we have half of the domain pairs in the twilight zone and the remaining half in the midnight zone. This is consistent with the fact that no single alignment, even the optimal, is statistically significant in this group. But the marginal probability over all possible sequence alignments is able to detect the relationship in some unspecified way. The final plot shows more than 80% of domain pairs fall in the midnight zone. For these pairs, even the marginal probability cannot assert a relationship. Then we did a qualitative case study. Here, we compare the sequence of human hemoglobin with sequences of 6 other related proteins at varying levels of sequence divergence. But all of them are classified within the same globin-like fold-in scope. The expected distance measure presented here takes into account the alignment represented in white and all the other alignments weighted by their posterior probabilities. The largest distance corresponds to the comparison of human hemoglobin with red algae phycocyanin. These sequences have diverged into the midnight zone of sequence relationship. Previous studies have noted that these proteins have an extremely distant evolutionary relationship and known to have no substantial sequence similarity, but similar folding patterns, something confirmed by these statistics. In sum, we have performed a comprehensive analysis of the compatibility of alignments derived from sequences and structures of protein pairs. This analysis identifies the amount of sequence divergence between pairs and the safe operating ranges of sequence alignments. First region is the daylight zone of sequence relationship. Here, the alignments agree closely with each other. Next, the twilight zone. Here, the alignments start to diverge, and any given residue level alignment should be handled with caution. And finally, the midnight zones of sequence relationship. In this region, any suggested sequence alignment generated from sequences alone should be discarded altogether. We have extended seq-malina for users to compute the expected distance between sequence and any given reference alignment. The QR code and the link is shown here. Thank you very much for your time.